Welcome back. This topic is on nervous tissue histology. Nervous tissue is composed of neurons and neuroglia. Neurons क्या है? Neurons are specialized cells that constitute the structural and functional unit of a nervous system. Neuroglia क्या है? Neuroglia are supporting cells. सो ये नर्वस सिस्टम के जो न्यूरॉन्स है उनको सपोर्ट करने के लिए जो सेल्स होते हैं वो न्यूरोग्लाया सेल्स रहते हैं. न्यूरॉन वी ऑलरेडी नो इट इज़ मेड अप ऑफ सेल बॉडी प्रोसेसेस. ओके, सो सेल बॉडी इज ऑल्सो नोन एज सोमा, प्रोसेसेस ये न्यूराइट्स. ओके, साइटोप्लाज्म यूजुअली शोज ग्रैन्युलर मटेरियल एंड दिस इज नोन एज Nissel's substance or Nissel's body. You can see here in the diagram Nissel's bodies or Nissel's substance. ये जो processes होते हैं there are many shorter processes. These are known as dendron or dendrites. Dendron का मतलब है tree के जैसे होता है, इसके लिए dendron. Whereas one long process is known as the axon. In dendron the nerve impulse travels towards the cell body. Under electron microscope, if we see here, these bodies actually have got rough endoplasmic reticulum, which shows that there is high level of protein synthesis in the neurons. And this is axon, if you can see here. ये अगर एक्सॉन देखोगे, ईच एक्सॉन का जो बिगनिंग है वहाँ पे निसल्स फ्री जोन बोलते हैं. वहाँ पे निसल्स ग्रैन्यूल्स नहीं रहते हैं. दिस पार्ट, ओके, निसल्स फ्री जोन, एंड दिस इज नोन एज एक्सॉन हिलॉक. साइटोप्लाज्म ऑफ द एक्सॉन इज कॉल्ड एज एक्सोप्लाज्म. इसका मेम्ब्रेन को कहते हैं एक्सोलेमा. द एक्सोप्लाज्म कंटेन्स सेल ऑर्गेनल्स ऑफ द न्यूरॉन्स ऑफ दैट इज ऑफ द सेल न्यूरॉन सेल बॉडी एक्सेप्ट राइबोसोम्स. इसका मतलब क्या है? प्रोटीन सिंथेसिस सेल बॉडी में होते रहता है एंड देन इट इज ट्रांसपोर्टेड थ्रू द एक्सॉन हियर, थ्रू द एक्सोप्लाज्मिक ट्रांसपोर्ट कहेंगे हम. ओके, एंड गोज टुवर्ड्स द एक्सॉनिक टर्मिनल्स. सो एक्सॉन हिलॉक क्या होता है? दिस इज अ प्लेस वेयर देयर इज नो निसल्स ग्रैन्यूल एंड हियर एक्शन पोटेंशियल्स आर जनरेटेड एंड देन दे आर कंडक्टेड अलॉन्ग द एक्सॉन. इतना से कॉर्नर में अगर आप देखोगे जहाँ पे एक्सॉन हिलॉक है और द इनिशियल सेगमेंट ऑफ द एक्सॉन यू कैन सी इट इज़ यूजुअली अनमाइलिनेटेड. इन माइलिनेटेड नर्व फाइबर्स हम बोलते हैं, उसमें भी ये इनिशियल सेगमेंट अनमाइलिनेटेड रहता है. दिस इज हियर वी आर सीइंग द न्यूरोफाइब्रिल्स. वन मोर डिस्टिंक्टिव फीचर ऑफ न्यूरॉन्स इज प्रेजेंस ऑफ नेटवर्क ऑफ फाइब्रिल्स. These are neurofibrils. Under electron microscope again, these fibrils are microfilaments, microtubules, neurofilaments. This part is the axon. Axon क्या करता है? It is conducting the impulse away from the cell body. So axon means nerve fiber. The bundle of nerve fibers found in CNS together are known as nerve tracts. ओके, ये काफ़ी सारे एक्सॉन्स अगर एक साथ दिखेंगे हमको, हम उसको ट्रैक कहते हैं. इन CNS, while in PNS, peripheral nervous system, we call it as peripheral nerves. The axon can terminate in two ways. In the central nervous system, it terminates by coming in contact with another neuron known as a synapse. Whereas outside the CNS, it can end in an effector organ like muscle gland, कहीं पे भी end हो सकता है, or it can synapse with the neurons in a peripheral ganglion. Okay, this is to show you the difference between axon and dendron. Axon single long process, multiple short processes. Axon is usually uniform diameter, the thickness can change as it divides, so it will keep on reducing. Axon is free of Nissel's granule, dendrites में Nissel's granule present. The nerve impulse in the axon is away from the cell body, whereas in the dendron it goes towards the cell body. The type of neurons are on the basis of the number of processes. If you see this is known as सिंगल प्रोसेस है ना, तो इसको यूनिपोलार कहते हैं. सिंगल प्रोसेस बाहर आ रहा है, उसके बाद ये डिवाइड हो रहा है. समटाइम्स इट इज नोन एज सूडो यूनिपोलर. ओके, हियर इफ यू कैन सी दिस इज बाइपोलर न्यूरॉन, जहाँ पे न्यूरॉन्स दोनों दो साइड द प्रोसेसेस आर कमिंग फ्रॉम बोथ द साइड्स, फ्रॉम टू साइड्स आई विल से. सो वन एक्सॉन है, वन डेंड्रॉन है. स्पाइरल गैंग्लियन इफ यू कैन सी. मल्टीपोलर न्यूरॉन मीन्स बहुत सारे, वेरी कॉमनली सी, बहुत सारे डेंड्रॉन्स दिख रहे हैं और एक एक्सॉन दिख रहा है. मोटर न्यूरॉन्स इज एन एग्जाम्पल ऑफ मल्टीपोलर न्यूरॉन्स. बाइपोलर न्यूरॉन का एग्जाम्पल हमने बोला न्यूरॉन्स इन द वेस्टिबुलर एंड स्पाइनल गैंग्लियन. whereas here unipolar का example is the neurons in the dorsal root ganglion. On the basis of function, we classify neurons as sensory neuron or motor neuron. Sensory neuron which carries impulses from receptor organ to the CNS, whereas motor neuron, it transmits impulses from the CNS to the muscles and glands. On the basis of the length of axon, we classify the neurons as Golgi type 1 and Golgi type 2. Golgi type 1 means long axon hota hai, which is connecting remote regions like pyramidal cells of motor cortex in the cerebrum. And Golgi type 2 जो होता है वो short axons होते हैं, वो नियर बाई ही एंड हो जाते हैं. स्टीलेट सेल्स, सेल्स ऑफ मार्टिनोटी ऑफ द सेरिब्रल कॉर्टेक्स, ग्रैनुलर सेल्स ऑफ द सेरिबेलर कॉर्टेक्स, ये Golgi type 2 है. Neuroglial cells. Neuroglial cells, we said they are supporting cells. They are much smaller in size than the neurons and they are much more in number. So number of glial cells can be approximately 10 to 50 times that of a neuron, and size is smaller than the neurons. In histological preparations, whenever we can see only the nuclei of the neuroglial cells. They are so small. And if you want to see the processes and each cell in detail, then they will require special techniques. Normally it will not be seen. And neuroglial cells in the central nervous system are astrocytes, oligodendrocytes, microglia and the ependymal cells. Whereas neuroglial cells in the peripheral nervous system are satellite cells and Schwann cells. Astrocytes. Astro means star shaped. So astrocytes are small star shaped cells which give off number of processes. These cells are present, they are two types, protoplasmic and fibrous. They are present for the nutrition of the neuron. So if you can see here in this right side diagram, this is a astrocyte cell and you can see these processes, how they are going towards the neuron. The processes of the astrocytes are united to those of the other astrocytes through gap junctions, and they communicate with one another through calcium channels. So they also play an important role in regulation of the synaptic activity, also in metabolism of neurotransmitters and neuromodulators. This process, if you can see the process of the astrocytes, they usually end in an expansion and they are related to the blood vessels. Let us see here. The astrocytes we said are of two types. There is fibrous, this one is fibrous, and the protoplasmic. Fibrous astrocytes are seen mostly in white matter. They are very thin and the processes are asymmetrical. Whereas protoplasmic astrocytes are seen in grey matter. The processes are thick. You can see in this diagram the processes are thick and they are somewhat symmetrical. Functions are biochemical support of the endothelial cells. They help in formation of blood-brain barrier, provision of nutrients to the nervous system, nervous tissue, maintenance of extracellular ionic balance, and also help in repair process by forming cellular scar tissue. they also serve as an insulator and they prevent neuronal impulse from spreading in unwanted direction. This is also very important feature. Then we come to oligodendrocytes. Oligodendrocytes, it has got a pear shaped body and very few processes. Oligo means scanty, इसके लिए scanty processes होते हैं, इसके लिए इसका नाम है oligodendrocyte. And these are the cells which provide myelin sheath to nerve fibers present in the brain and spinal cord. तो अगर MCQ में पूछ सकते हैं कि myelinations of the nerve fibers in the central nervous system is formed by, it is the oligodendrocytes. And peripheral nervous system में क्या होता है? Which cells form? The Schwann cells. So myelination is by Schwann cells in peripheral nervous system and oligodendrocytes in central nervous system. Very important. Oligodendrocytes are present both in grey matter as well as white matter. Ependymal cells. Ependymal cells are, these are the cells which line the ventricles of the brain and the central canal of the spinal cord. ये low columnar epithelial cells रहते हैं ये, they are lining the ventricles of the brain, we said. These cells secrete cerebrospinal fluid. In some regions they may have got cilia, so help in movement of the CSF. The ependymal cells, we said, they line the floor of the ventricles, especially the floor of the fourth ventricle where it is known as tanycytes, and they have got long basal process. So what is a tanycyte? Tanycyte is, these are the ependymal cells which line the floor of the fourth ventricle and they have long basal process. Function वही है, secretion of cerebrospinal fluid. They also help in maintaining the chemical composition of the central CSF, cerebrospinal fluid. Next cell is the microglia. Micro means chota. Okay, small cell, a small supporting cell. Okay, so they usually phagocytose the cellular debris, means they are the membrane of, they are the members of mononuclear phagocyte system. Mononuclear phagocyte system, ये अपने को help करने का काम करता है. Okay, it is mesodermal origin. They are seen more numerous in grey matter than in white matter, but present in both. तो ये क्लीनिंग का काम करते हैं. They act as a phagocyte, they clear the debris, damage, जो कुछ भी damage structures होता है, and they usually become active after there is a damage to the nervous tissue by trauma हो या disease. Also they protect the nervous system from viruses, microorganisms. So it is a part of the mononuclear phagocytic system in CNS. Microglia, they are the smallest neuroglial cell present in both grey and white. They are also known as scavenger cells. So they form the brain's immune system. So we have seen neuroglial cells in the central nervous system. Neuroglial cells, smaller in size than the neurons, more in number, 10 to 50 times more than the neurons, and astrocytes, oligodendrocytes, ependymal cells and microglial cells. This is what we have seen. Now let us see the neuroglial cells in the peripheral nervous system. Peripheral nerves, we know, that is bundles of axons together outside the CNS are known as peripheral nerves. They can be myelinated or unmyelinated. These nerve fibers, they carry impulses from the spinal cord or the brain to the peripheral structures. Okay. These fibers, of course, they are called efferent or motor fibers. Efferent fibers are axons of neurons located in the grey matter of the spinal cord or brain stem. Nerve fibers carry impulses from peripheral organs to the brain or spinal cord. They are known as afferent fibers. Afferent nerve fibers are processes of neurons that are located in the ganglia. This we just revised, okay, about the efferent and the afferent fibers. Let us see the neuroglial cells. The neuroglial cells in the peripheral nervous system are satellite cells और कैप्सुलर सेल्स. क्यों? क्योंकि ये सराउंड करता है the neurons of the it surrounds the neurons of the ganglia, satellite, or the, you can see here, the capsular cell. So they are flat cells, they have a very prominent nucleus, they surround the neurons of the spinal or the autonomic ganglion, and they form a multinucleated capsule. So capsule अगर देखोगे तो वो multinucleate दिखेगा, as we said ना, they have a very prominent nuclei. So they support and protect the neuron. Satellite cells, these are the principal cells in the PNS, principal glial cells in the peripheral nervous system. Okay. They are, if you see much more in detail, they are derived from the neural crest. They supply nutrients to the neurons and also act as a protection or cushioning cells. Next, another important cell, Schwann cells. Schwann cells are responsible for formation of the myelin sheath in the peripheral nerves. So Schwann cells are flattened cells, they have a flattened nucleus. They cover the nerve fibers and this is known as the myelin sheath. So any axon can be myelinated or unmyelinated. Myelinated अगर होगा तो myelin sheath अगर जिसको होगा उसको myelinated axon कहेंगे. So myelin sheath, it is an insulating sheath which is surrounding the axon outside the axolemma, and this sheath or myelin sheath is formed by the Schwann cells in peripheral nervous system and oligodendrocyte in the central nervous system. We are repeating this because this can come as a MCQ. Okay. Many Schwann cells can surround the axon to form a myelin sheath in peripheral nervous system, but a single oligodendrocyte provides myelin sheath to many axons in the CNS. Okay, that is the difference. So let us see here in this diagram. We can see the myelin sheath. This is a sheath and this is the axon. So how the axon, it is taking up the axon. I think I have one more diagram which can explain, self-explanatory. Yes. This is the axon which is lying near a Schwann cell. It enters the Schwann cell. See here, it is invaginating, and this part is known as the mesaxon. Now, with this mesaxon, with this, it actually suspends this axon into it, and it starts on curls round. Or this is, see how it is starting winding around the axon, and this is one nucleus. So this is how a myelin sheath is formed. So this slide shows formation of myelin sheath. An axon which is lying near the Schwann cell is invaginated into the Schwann cell, and in this process it is, that is, the axon is suspended by a fold known as the mesaxon. Now what happens is this axon goes on winding around the this Schwann cells goes on winding around the axon because the axon is suspended, na? And what you see is the final outcome, that is the axon being surrounded by a myelin sheath. There are several layers. See, you can see there are several layers of this Schwann cell forming here. One Schwann cell. Huh? This slide is showing the myelination of the axon or myelinated nerve fiber. This is the myelinated or the Schwann cell which has myelinated the nerve fiber, and इसके बीच में जो ये गैप दिख रहा है इसको कहते हैं nodes of Ranvier. So nodes of Ranvier are very important, physiological importance. मतलब the impulse is traveling down the nerve fiber, it usually jumps from one node to the other node, and this is known as saltatory conduction. So fast conduction होता है, और unmyelinated neuron अगर रहेगा तो impulse will travel along the axolemma, and that will be a very slow as compared to this. Okay, so this conduction, as we said, will be slower than the saltatory conduction. This is it, in between the Schwann cell I can see what is known as the node of Ranvier. This is very important, asked in your viva as well as theory. Myelin matlab it contains proteins, lipids and water. This is to show you about unmyelinated axon. Like we can see here, this is one Schwann cell and there are so many axons nearby. वो invaginate हो जाते हैं, that's all. उसके बाद वो सस्पेंड होकर उसको वाइंड करने के लिए जगह नहीं है. ओके, सो दिस इज वेयर one Schwann cell is taking up so many axons is known as a unmyelinated axon. That's all. The mesaxon does not form, that does not spiral around it. Okay, see, this is a Schwann cell, everywhere it is seen. Here you can see myelination, and the left side of the diagram shows node of Ranvier and how myelination is done here, and this side right diagram is showing you that there is unmyelinated axon, many axons invaginating into one Schwann cell. This slide shows the difference between myelinated nerves and unmyelinated nerves. Myelinated, the axons are of large diameter, axons are of smaller diameter in unmyelinated. In myelinated, the axons surround by the concentric layers of the Schwann cell plasma membrane. Here, axons are surrounded by the cytoplasm of the Schwann cell because many axons are invaginating. Single axon is surrounded by many Schwann cells in myelinated nerve, whereas in unmyelinated nerves, a single or a group of axons are invaginated by a single Schwann cell. In myelinated nerve, which is this point is important, the nerve impulse jumps from one node to other, and this is known as saltatory conduction. So the conduction is fast. Whereas in unmyelinated nerve, the nerve impulse travels uniformly along the axolemma, and the conduction is slow. Now we will see the microscopic structure of peripheral nerve. ये अगर एक पेरिफेरल नर्व का हमने अगर सेक्शन लिया तो वी कैन सी दैट द नर्व फाइबर्स आर अरेंज्ड इन फॉर्म ऑफ बंडल्स एंड आर सराउंडेड बाय कनेक्टिव टिश्यू. दिस इज वन एक्सॉन, माइलिन दिखा रहा है यहाँ पे. दिस नर्व फाइबर इज कवर्ड बाय एंडोन्यूरियम. एंडोन्यूरियम कंटेन्स टाइप 2 कोलेजन फाइबर्स. These nerve fibers together form one bundle or a fasciculus. If you can see, this is one fasciculus. This one fascicula is covered by perineurium, and there are so many such fascicles together, which is together covered by, on the outer side if you can see this, which is known as the epineurium. So nerve fiber is surrounded by endoneurium. Many nerve fibers form fascicle. Each fascicle is surrounded by perineurium, and many fascicle together is surrounded by the epineurium. Okay, so entire nerve, what you see is one nerve, and it is surrounded by epineurium. So if you take a TS of a peripheral nerve and under slide, what you will see is in the right side diagram, you let us see schematically represent किया है ये. You can see a cell nerve fibers. You can see ये myelinated है ना, इसके लिए मुझे एक राउंड दिख रहा है और बीच में एक डॉट दिख रहा है. डॉट मतलब वो एक्सॉन है. वो राउंड स्टेन नहीं हुआ है बिकॉज इट इज़ अ माइलिन शीथ. सो माइलिन इज गिविंग दैट एम्प्टी अपीयरेंस. ओके, एंड of course that we'll call it as endoneurium, and this is the fascicle को surround किया है ये perineurium, and this entire nerve fiber I can see here covered by epineurium. So points of identification are the each nerve fiber appears to be empty with one dot in the center, which is the axon. Emptiness is seen because of the presence of myelin. Then we can see some Schwann cell nucleus. Of course Schwann cell का न्यूक्लियस इधर उधर दिखता है, बीच में ये जो डार्क दिख रहा है स्टेन ये. Then we will discuss about here in the Viva slide. Will tell about the epineurium, endoneurium and the perineurium. One more slide to show you the transverse section. These are many fascicles together. This is one fascicle again, which is taken as a close up to show you all how you can see the axon, the myelin sheath, and certain dark blue color nucleus जो दिख रहा है, that is the nucleus of the Schwann cell. Another schematic representation on the left side. Right side, this is how you can draw in your histology journals. Epineurium, perineurium and endoneurium. This is LS of a nerve fiber and here special stain, that is the osmic acid stain. Osmic acid stain इसके लिए लिया है क्योंकि वो माइलिन शीथ को स्टेन करता है. ओके, HNE में क्या होता है, माइलिन इज वॉश्ड अवे, वी सेड, एम्प्टी अपीयरेंस दिखता है ना. तो यहाँ पे ऑस्मिक एसिड विल स्टेन द माइलिन शीथ एंड वी कैन नाइसली सी दिस नोड्स ऑफ रैनवियर्स, प्रॉपर डिप्रेशन दिख रहा है यहाँ पे. ओके, सो दिस इज अ LS ऑफ अ नर्व फाइबर स्टेन्ड बाय ऑस्मिक एसिड. Why osmic acid? Because osmic acid can stain the myelin sheath, and here myelin sheath is being stained black. One more slide to show you the peripheral now. TS, this is the high power. Okay, now we will discuss about the ganglia. Collection of neurons outside the central nervous system is called as ganglion. This much is all about the peripheral nerve histology as well as general anatomy of a nerve. Thank you.